Hello and welcome to another episode of the Revisited series, Season 2. In this episode we'll cover lentils again. We'll go over the domestication and breeding of lentils. Lentils were first domesticated in the Fertile Crescent of the Near East. From there it spread to Europe, the Middle East, Northern Africa, and the Indo-Gangetic Plain. The domesticated and wild subspecies of Lens culinaris, the species named for the common lentil, is found in the Middle East, specifically in the Fertile Crescent. This species, in terms of its domesticated form, is as old as einkorn, emmer, barley, and peas, and is likely to be one of the earliest domesticated pulses produced and grown. It is also estimated that this crop was domesticated 8,000 years ago. The wild species is indigenous to southwestern Asia and the Mediterranean region. As of right now, it's propagated and produced in most of the subtropical and temperate regions within the world, particularly where rainfall is low since it likes a drier climate. Most of the germplasm of lentils are classified in three categories: subtropical savanna, Mediterranean, and northern temperate. It is also classified in two cultivar groups: the large-seeded macrosperma group and a small-seeded microsperma group. The former is cultivated mostly in Europe, North Africa, and the United States and Canada, whilst the latter is cultivated mainly in Asia, Egypt, and Ethiopia. Western Asia and southeastern Europe grow both varieties. Based on the work by Dr. Hellbeck, it is likely that the history of the lentil is as ancient as agriculture itself. The oldest known remnants of lentil date back to the Pharyngi cave in Greece and are dated to 11,000 BC. The domesticated lentil and its wild relatives are all self-fertilizing diploids in nature, with a total of 14 chromosomes, although the chromosome lengths vary from species to species, preventing crossbreeding in most instances, except in the cases of the wild subspecies and domestic subspecies of Lens culinaris. The gene pool of lentils is divided into three groups: the primary, secondary, and tertiary gene pools. The primary gene pool is Lens culinaris, which includes the cultivated forms and land races and its wild ancestor. The secondary gene pool are two species, Lens eryvoides and Lens nigricans. These two species can crossbreed with the common Lens culinaris species, although with great difficulty. The tertiary group consists of all the remaining Lens species, which are incredibly difficult and not impossible to crossbreed with, since most of the progeny die or become sterile. The wild subspecies is commonly used to breed varieties of lentil that are far more cold tolerant. They also provide traits such as drought resistance, wilt resistance, and astrocyta blight resistance. Crossbreeds with Lens nigricans and Lens culinaris can form hybrids, but the resulting hybrids have low levels of seed. Now onto the breeding objectives. Within lentils, the breeding objectives vary from location to location depending on the region of the world. The key goals of exporting countries when it comes to lentil are higher and stable seed yield, high disease resistance, and better seed quality. While in import-dependent countries such as India, a general increase in yield per hectare is a primary aim. As a general rule, the lentil breeding program always involves incorporating different traits from diverse lineages to increase traits of interest, as well as create new genetic combinations to increase yield and stress resistance. The wild ancestor of the common lentil, Lens culinaris subspecies orientalis, is much smaller than its domesticated form, and have seed pods that burst immediately upon maturation. It is from this wild ancestor that the land races were derived, followed by the development of pure line selection within heterogeneous land races, which to this day is still the most common method of cultivar development during the early phases of lentil improvement. However, in modern days, hybridization and selection have become the chief means of lentil improvement in the past 3 to 4 decades, especially in the developed world. The breeding process is much akin to soybean breeding, as well as the methods involved in most found in self-pollinating crops. See the video in the description for more details. Most of the key traits such as disease resistance, yield, and abiotic stress tolerance can be found within the lentil gene pool. But as for other traits, there is scarcity in the genetic variability, which represents a limitation for lentil breeding in the current era. In terms of breeding these plants, it is quite difficult since the methods of artificial hybridization have a success rate between 20 to 50%. This is because, unlike most other legumes, the flowers of a lentil are very tiny and fragile. The process of emasculation, which is the process of removing the pollen parts to prevent self-fertilization, and the process of adding new pollen from another plant onto the emasculated flower, usually cause injury to the floral parts, reducing success rate. These problems are managed by selection of larger flowers on the plant, using lateral buds instead of terminal ones. A far more careful method of emasculation and pollination is required, and it's been found that lower temperatures actually improve fertilization. Although high temperatures in the early morning also seem to be successful, so the best time to fertilize is early in the morning. Parent selection is also a major choice within hybridization. The most common cultivars selected are those with a wide ability to adapt to different climates, higher yield, and disease resistance. Other attempts to breed new varieties by integrating exotic genes from other species that cannot naturally crossbreed with the common lentil have also been done using somatic hybridization, or the fusion of two cells together in a tissue culture medium before growing out a plant resulting from those two fused cells. The successes of this process have been very limited as of right now. Mutation breeding has also been utilized in the breeding of lentils. Indeed, you can see some experimental varieties derived from these mutations on the screen right now. Two of the most popular cultivars, Ratjan and Ratjendra Masura 1, were derived from mutations created through mutagens applied to seeds. The mutations allowed for improved traits such as higher yield and spreading type, as well as tolerance to low temperatures. Other mutations that showed up were early maturity and suitable for late sowing mutations. As of right now, there's 453 mutant legume varieties worldwide. Most of them were released in the 80s, but none have been released since. That mutant that has that spreading trait has been utilized heavily in breeding new varieties of lentil since its creation, however. And that about covers everything. Thank you for watching. Thank you for watching this video. If you're watching on YouTube, please subscribe to me on BitChute for a greater variety of content, 4 videos a week. And thank you to all my subscribers on both platforms. I appreciate it.